So the topic for today really focuses on biomarkers and early cancer detection. I'm going to digress a little bit from that, as you'll see as I move forward, moving on from simply early detection to try to understanding issues with prognostic capabilities of biomarkers, and then I'll end up with some new technologies and new developments. So I thought it's a good place to begin is trying to define the topic. So the definition here for a biomarker, I lifted actually from Wikipedia, which is a place where I often go now for curated or semi-curated scientific information. So the Wikipedia definition of a biomarker is a substance that's used as an indicator of a biological state. It's obviously a very large definition, but it's something you can objectively measure, and you can evaluate it as an indicator of either a normal process, a normal biological process, a pathology, or in the cases of some of the things I'll talk about a bit later, a response, a pharmacologic response to a therapeutic intervention. Once you have a biomarker validated, it can be used for a variety of purposes. It can diagnose disease risk. It can determine presence of disease in an individual, and that's where we focus here on early detection. And then hopefully when we emerge into the era of personalized medicine, it can define or tailor treatments to a specific disease, to a specific tumor type, or to a specific individual, as well as the type of administration regime. So this slide is actually from a review published a few years ago that I think nicely articulates a number of these different areas where biomarkers can be used. So it's an example from breast cancer where you may have a biomarker of assessing early risk. So this would be like BRCA1 or BRCA2 indicating some familial risk. A biomarker for screening, which in this broad context of biomarkers we could call mammography as fitting into this pigeonhole. And then looking at biomarkers indicative of risk, for example, ER or PR or HER2 expression, and then in the future biomarker predicting recurrence, so you could try intervening at an early state. So there are many different points on this continuum, if you will, of tumor development where biomarkers can be quite useful. So what I'm going to focus on for the next roughly 40 minutes or so, I'll emphasize quite a bit of work in biomarkers in early cancer detection and emphasize why that could revolutionize cancer care. I'll also focus a bit on biomarkers in predicting recurrent disease or cancer outcomes. And then I'll end up talking a little bit about some ways to identify novel biomarkers using proteomic-based methods and mouse models systems. So Lee Hartwell at the Fred Hutchinson Cancer Center has been on a crusade for roughly the last 6 or 7 years of really pushing the idea of early detection as a way to impact the morbidity and mortality due to cancer. So this was a review published in Nature Cancer Reviews several years ago that in some ways threw down the gauntlet, saying that there's a tremendous amount of funds invested in research targeting treatment. However, if you look at the epidemiology and look at cancer outcomes, you have the potential to make the greatest impact in early detection. And here's an example from this paper. So it's a little bit blurry, I apologize. What you can see on the y-axis is the survival rate, 100% to 0%. And on the x-axis is the stage of disease when it's identified. So on the far left is diagnosis when the cancer has already metastasized or distant disease, regional diagnosis, and then on the right here is when the cancer is diagnosed locally, so no evidence of spread. And what you're looking at then is survival rates, 5-year or 10-year survival rates for breast cancer, colorectal cancer, lung cancer, and prostate cancer. Now, each of the bars, the different colored bars, represents a period of about 4 years spanning several decades in time. So the very light pink on the far left is in the mid-70s, and then in the blue color, more on the far right, we're down into the late 90s. So a couple of important points from this. One, you can see that the survival rates are clearly related to when the cancer, at what clinical stage the cancer was diagnosed. So for breast cancer, the 5-year survival rates, if you're diagnosed with distant disease, when you present, about 20% of 5-year survival. Whereas if you're diagnosed with local disease, you have roughly 80 to 90% survival. So that's an important point here. The second important point is that the change in terms of survival really hasn't demonstrably changed over time. In other words, if you were diagnosed back in the late 70s with distant disease versus diagnosed more currently in the late 90s with distant disease, your chances of survival only incrementally improved. Same with regional and with early disease. So the key here is that diagnosing it early before there's evidence that the cancer has left the primary site is the optimal time for cure, and that's in many ways quite intuitive. So from the same paper, these investigators estimated what impact you would have on survival if you did diagnose most of these cancers at the local primary disease state. So if currently about 40% of colorectal cancers are localized when they're detected, and the 5-year survival rate for all comers here is about 64%. Yet if you diagnosed everyone with localized disease, your 5-year survival rate would be 90%. So you get the idea of where we're going here. Now, for a biomarker to actually make it all the way into clinical use, there's a number of hurdles it needs to pass. And so this is a schema from this same review article, but really based on some work by Margaret Pepe here at the University of Washington. And that is these 5 phases, and I would argue there's probably actually a 6th phase. But these 5 phases are: you initially identify a marker from preclinical or exploratory-based studies, and I'll give you some examples of those later on in the talk. You then develop your clinical assay, and you develop this as a test, which you would hope to use actually in clinical practice. Then you evaluate this test on some type of retrospective study sample. Then you move on into a prospective screening study, and then finally, to determine whether it actually does control cancer rates, you need to do a prospective population-based study to see, does that test reduce the population burden of the disease. And so these are the 5 steps, and most of these biomarkers that are evaluated from these early studies, they really fail right after this retrospective study. Many of them work reasonably well in retrospective studies, but they really don't make it through a prospective screening study, and I'll describe a couple of those in just a moment. Now, the 6th phase, I would argue, for some cancers, you don't even want to know actually that they're there because there's not much of an impact on simply detecting that cancer, and prostate cancer is a prime example, and I'll spend a little bit more time on that. So I would argue you don't want to simply determine does the test reduce the disease or the population burden of the disease. You really need to determine, does it reduce mortality or morbidity of that disease. So the successes here of biomarkers that have made it through this entire decathlon or gauntlet are very few. So here's 4 examples, and of these, really only the pap smear for cervical carcinoma has made it all the way through that pipeline, definitively shown to reduce cancer-related death. I think mammography, in some cases, I would say it's really plus minus at this point. Fecal occult blood for colorectal cancer, that's a no. And for PSA for prostate cancer, it's arguably not yet. But there's still hope, although it's been more than a decade since that marker has been out there. But many biomarkers can assist in diagnosing cancer. So this is an entirely different scenario than population-based screening for identifying cancer. This is when you already have some clinical suspicion of the disease and you want to confirm that particular diagnosis. So we have many, many examples. And here's a table of FDA-approved biomarkers that have been evaluated and vetted for a number of these indications, and the ones with the yellow arrows are ones that have been approved for screening. And here we're back to the pap smear and PSA. These last 2 down here are for bladder cancer, and I think it's arguable, I don't know anyone that actually uses these as a screen for carcinoma of the bladder. So out of this entire list here of FDA-approved biomarkers for cancer screening, there's really basically 2, and PSA remains controversial. So why is this so challenging? What's the problem? And I think there's two main problems here. One is that, if you really think about it, neoplastic cells or carcinoma cells are actually quite similar to their normal counterparts. There are a number of genetic changes at the genome level, at the transcript level, at the protein level, but they're actually very few. If you, for example, do a gene expression array analysis to look for transcript changes, the vast majority of transcripts are the same in terms of quantitative levels. So that's just one example. Clearly different than infectious disease, where you have diagnostics. Those genomes, those organisms are entirely different, and vast majority are quite different than the host. The second problem is that by definition, we're talking about early markers, the disease burden is vanishingly small, so the product of those cells that you would try to assay in a biological fluid is going to be very, very small. So you're really searching for that proverbial needle in a haystack. So here's an example actually from some beautiful work that was published by your next speaker. So you have Lee Anderson coming next week. He's really a true leader in the field of proteomics. So this is from his Plasma Proteome Institute. And he is one of the leaders in the field to simply define basic abundance levels and diversities of proteins that are present in the serum or plasma. And so here's the problem. So if you look at all the proteins in blood, 90% of them, if you just look at the absolute quantity, are comprised of only 10 proteins, albumin being the the gorilla on the block here in yellow. Of the remaining 10%, an additional 9% is made up of 12 proteins. So 22 proteins account for 99% of all of the mass of proteins in the blood. So you're left with 1% here that is what you're looking for a protein-based biomarker. So it's a daunting task in terms of a discovery-based experiment to try to find biomarkers of cancer in this. The second problem here is this tremendous dynamic range. So again, from Lee Anderson's work, you can see that the classical plasma proteins are present here in log differences relative to some of the classic tumor markers that we currently are evaluating. So you're looking at log level differences, orders of magnitude of 10 to 12, which is a huge dynamic range. So PSA is hiding right down here. Here's some of the inflammatory markers, the interleukins. So you can definitely measure them by sensitive assays like ELISAs, but identifying them or discovering them is really quite challenging when you have all of this other noise to worry about. Now the second important aspect in thinking about biomarkers is tackling the problem of sensitivity and specificity and the positive and negative predictive values. And I think probably many of you are well versed in this type of a table. Here's an example from colorectal cancer, emphasizing why fecal occult blood for detecting colorectal cancer is a very poor biomarker. So in this example, the true positive is 2, the false negative is 1, so your sensitivity is only around 60 to 70% of diagnosing that cancer, whereas the specificity, one might argue that 90% specificity is reasonable. However, since you're screening, you would potentially be screening millions of individuals, having this level of false positives is way too high because it leads to many other tests, which are in turn invasive, expensive, et cetera. So again, one of the reasons why fecal occult blood testing simple to do, but it doesn't meet the metrics of the type of a biomarker that you would want to sensitively and specifically identify a cancer. Now, the positive predictive value also suffers from a similar problem in the sense that it's not intrinsic to the actual test. It really depends on the prevalence of the disease in the population. So you could dramatically improve the positive predictive value of fecal occult blood if you were screening, for example, individuals with a family history of colorectal cancer as simply one example. But you're dealing with population-based type of studies. So let's consider PSA as an example of this in the context of prostate cancer. So PSA was discovered in the 70s and purified as a protein in the late 70s. It's a serine protease that's secreted or produced by normal prostate epithelial cells and also secreted and produced by prostate cancer cells, actually at a lower level on a cell-by-cell basis than benign epithelium, but it is exquisitely prostate specific. It's not really produced by any other cell in the body. It does complex in the blood with multiple other proteins. There are variants of PSA that may possibly provide some discriminatory value from a cancer versus a benign process, but this really hasn't made a lot of inroad into clinical medicine. But beginning in the 80s, PSA was starting to be used as a screening test for prostate cancer, since it was known that men with very advanced disease had very, very high levels of PSA. The idea was, could you use some type of a cutoff to discriminate cases of prostate cancer much earlier in the disease course? So when it was originally approved, it was really proposed as simply a dichotomous variable, and a cutoff of 4 nanograms per deciliter was used as the discriminatory range. So less than 4, in theory, no cancer; greater than 4, suspicion for cancer, go in for a biopsy for confirmation. I should say that it's an excellent, outstanding marker for detecting cancer recurrence once you've resected or removed the prostate gland, since PSA shouldn't be produced by any other tissues. Any level of PSA present after the prostate is removed indicates you still have a prostate cancer there somewhere in the host. Now, if you look at the actual performance of PSA, this is what's termed a receiver operating characteristic curve, or an ROC curve. The perfect biomarker would fall on this red line. So it would have absolute or 100% sensitivity and 100% specificity. This is how PSA responds for all comers. Just focus on this line. So you can see at no point on this line is there any sort of reasonable cut point where you would argue you had sufficient sensitivity and specificity. There's been a lot of work trying to make these cut points finer by discriminating according to aggressiveness of the cancer, the Gleason grade, et cetera. However, it actually remains a fairly poor marker. And this is a great example from some beautiful work that came out of the prostate cancer prevention trial that was performed several years ago, a trial that was using a drug called finasteride to prevent prostate cancer. This is from the control arm for men that did not get any drug. These biospecimens that came out of this study have been extraordinarily useful for trying to understand the performance of a biomarker like PSA. So what I'm showing you here are about 3,000 men on the control arm who had PSA values measured, and all of these men then subsequently had a prostate biopsy to determine, do they have cancer in the prostate gland. So if you look at the men that fell into each of these PSA cut points, so less than 0.5, less than 1, all the way up to 4, and they stopped at 4 because that was actually the study entry. No one greater than 4 was actually entered into this study, so you have a ceiling here. So here's the number of men at each of these points, and the percentage or the numbers of men at each of these cut points that were actually found to have prostate cancer in the prostate. So of 486 men that had PSA levels of less than 5, 6% of them had prostate cancer in the prostate. So there's no point here, if you look at the sensitivity and the specificity, where PS actually works very well in excluding prostate cancer or absolutely ruling it in. So it's still a test that has many problems. One of the reasons it may work as well as it does is because of the prevalence of prostate cancer in the population. And I'll get to this in a little bit more detail, but basically, if you start to biopsy prostate glands, you are generally going to find cancer. Certainly that prevalence increases with age, but that is one of the reasons, and Dr. Stamey at Stanford published many papers saying that that PSA was simply reflecting benign growth of the prostate, And again, the reason you're diagnosing more of these cancers is simply because you're putting needles in there and you're going to find a malignancy that approaches 100% prevalence in the population. So a few things about prostate cancer. There are about 180,000 new clinical cases diagnosed every year. About 50% of all men over age 50 have foci of prostate cancer if you start to biopsy. If you look for it, you'll find it at roughly that at that rate, which means there's more than 20 to 30 million American men with prostate cancer that remains undetected, and I think that's actually a good thing. However, well, then because of these numbers, most men actually die with prostate cancer than because of prostate cancer, yet we still have about 30,000 men a year that die of prostate cancer. So what we really need are biomarkers, whether it's a blood-based biomarker or a tissue-based biomarker, that can provide some information to determine risk of progression. We want to distinguish those cancers which will remain latent for decades versus which of those have the propensity or the opportunity to progress and become lethal. So here's a study, an autopsy study, that simply emphasizes this point of the latent cancer problem. And this was starting at age 50 up through 80. These are men that had autopsies done after they died of some other cause other than prostate cancer. So these men were not known to have any evidence of prostate cancer, and the prostate gland was looked at. So you can see that, and these numbers are actually probably underestimates compared to other studies, but about 20% of all 50 to 59-year-olds, 36% of all 60 to 69-year-olds, et cetera, have prostate cancer in the gland. There was actually another study, an autopsy study of men 30 to 49 years old, so 30 to 49-year-old individuals, 30% of them had cancer in the prostate. So we rarely ever diagnose clinically someone with in that age range with prostate cancer. It does happen, but it's certainly not common. So this prevalence issue is a major problem. So once you're diagnosed, there are many options for prostate cancer in terms of treatment. Watchful waiting or active surveillance is an approach that's gaining momentum, and I'll talk about that at the very end of the talk, but it sounds very counterintuitive for any malignancy that you would simply watch it. I think that actually may be important for other cancers as well, but prostate cancer clearly, based on the epidemiology, warrants this consideration of this approach. Surgery, radical prostatectomy, external beam radiation, brachytherapy, all these are options that are widely used. The key question is, are these cancers organ confined when you're evaluating the patient? Has it already spread, or will it never spread? And we need biomarkers that can distinguish these. Now, this idea of active surveillance is really brought home by this study. There have been two updates published following this, and this is radical prostatectomy versus watchful waiting in a population. So these are men in Sweden that were diagnosed with prostate cancer by prostate biopsy. They were randomized either to simply watch them or to have surgery to remove their prostate cancer. So you can see out now to 10 years, here's the follow-up, and the cumulative incidence of death due to prostate cancer is shown here up to 100%. So you can see that the men that were treated, that had surgery, they have about a 10% death rate over 10 years dying of their prostate cancer, whereas those men that were untreated, no surgery, have about an 18% or so death rate due to prostate cancer. So what that means is you have this number of patients, of men, exceeding 80% that could avoid any sort of therapy. You don't even want to have them diagnosed, actually, because of the natural history of the disease. There's this group of men here in the middle that do benefit from treatment, and then there's a number here who died despite curative intent. So these are individuals that had micrometastases early on that would benefit, we would hope, from more aggressive therapy at the beginning. So understanding and identifying these potential markers for disease progression is key. Biomarkers of disease presence, of disease virulence, as I mentioned, and then hopefully distinguishing the type of therapy that would optimally be given to those tumors for response. So where this is leading is to much more individualized therapy that treats tumors as being different, the host as being different, and then the actual agent that you're using as being different. So getting into some strategies for identifying these biomarkers. And where I'm going to focus a little bit here for the next few slides is on using more high throughput technologies and methods to identify distinguishing characteristics of benign cells and neoplastic cells, with the ultimate intent that these fingerprints or individual genes or proteins could be used in such a fashion for predicting outcome. So the underlying principle behind even using this type of a strategy is that the genes that are utilized or expressed from a particular malignant cell really dictates the phenotype. That is, it underlies the ability of that cell to have a malignant potential to metastasize or to resist or survive chemotherapy or radiation. And so by assessing this inventory of genes or proteins that are expressed, you can then develop that into a predictive marker. So the idea is that these truly would represent a fingerprint, that is, being something that's unique that associates with the disease state. So I'm not going to get into all the details of microarray-based studies. I'll just walk you through basically what we're just simply depicting here in these cartoons. So each of these spots represents a different gene. The color intensity represents how great it might be utilized or expressed. A green dot means it's more highly expressed in a benign cell. A red dot means it's more highly expressed in a neoplastic or a malignant cell. So the idea here is that at patient entry, when you first would see this individual, they would have a tumor biopsy, and a profile would be performed on that tissue by transcript profiling, in this case by microarray analysis. And depending on what that profile showed, you might observe that individual. They might be offered some other type of therapy, either something as simple as diet or nutrition or more complex, or they may need some type of more aggressive therapy. And then you would reevaluate their tumor after that therapy to determine a response or a progression type of signature. Now we have one example, or actually a couple examples in prostate cancer where this can be quite useful. So we do have a grading system in prostate cancer called the Gleason grade, which does afford some prognostic capabilities. It's probably actually the best indicator right now that we have how a given tumor will actually behave. And arguably it's a molecular marker in the sense that those features of the malignant cell or the patterns of those glands are dictated by the gene utilization or alterations within those cancer cells, all the way from a very well differentiated cancer where you're still forming glands through to individual cells that are invasive. So we set out a couple years ago to try to define a molecular correlate, a molecular underpinning of what the pathologist is actually calling a particular grade. Now what I'm showing you here are the results of a gene expression analysis of benign versus cancerous prostate tissue from about 30 individuals. So each column here is a different person, different patient sample, and each row is a different gene. On the far left are low-grade cancers, and on the far right are higher-grade cancers. So what I'm showing you here are gene expression changes that occur in generic prostate cancer. So these are very consistent changes across all prostate cancers that we looked at. So we are not intrinsically interested in these because there are many low-grade cancers, in fact probably most low-grade cancers, that will never progress, as I discussed earlier based on the autopsy studies. However, they are interesting in terms of understanding the biology of generic prostate cancer. What we're much more interested in is understanding those changes that occur with specific cancer grades. So Larry True, who's in the audience, performed this study where he microdissected the individual Gleason patterns, so Gleason pattern 3, pattern 4, and pattern 5, and then performed this transcript profiling analysis to determine, well, what gene expression changes associated with cancer grade. So you can see that there are clearly a large number of genes that discriminated the Gleason pattern 3 cancers, low-grade cancers, from the higher grade cancers, pattern 4 and 5. We couldn't distinguish between pattern 4 and 5. There were no genes that would clearly demarcate those higher grade cancers, but we clearly could discriminate between low Gleason pattern 3 and the pattern 4/5s. I'm not going to go through a lot of the details about this study, only to say that you can then confirm these changes at the protein level, which is at a point that's much more amenable to more traditional pathology or lab medicine based assays. So 2 of the genes that we identified, monoamine oxidase A, clearly increased in expression at the protein level with grade, as did a gene called defender against death, an anti-apoptotic protein. And we're now looking to see, can we find these actually in the blood of individuals with men that correlated with the different grade, since that's ultimately where we want to go, is something that could predict aggressiveness of the cancer, not simply the presence or absence of prostate cancer. Now, these particular profiles have been, you've probably seen many reports of them being studied in the context of outcomes. So here's was probably the original paper putting this whole concept on the map by Lou Stout's group back in the year 2000, and they had molecular signatures that could predict outcomes of different types of B-cell lymphomas, even lymphomas that looked histologically indistinguishable. They could find prognostic signatures, and that's been carried out carried through in a number of other studies, and I'll show you a couple examples of in the prostate in just a moment. However, it's very important to recognize that these types of biomarkers can be influenced by factors that we have no real control over at this point, and many times we're not even looking for it, yet we should. And I'll give you one example of this. So this was a study that we did to see what kind of influence simply diet would have on a biomarker in the prostate gland. So we did the same type of study. In this case, these are men who were enrolled in a dietary study of a very low-fat diet for 4-month period of time versus their own normal kind of high-fat American-based diet. Dan Lin, a urologist here at the University of Washington, biopsied these men before study entry and then repeated the prostate biopsy after simply this 4-month of diet. And you can see the gene expression changes that occurred very consistently simply due to diet. So in many of the studies that you see published, I think almost none of them that I can recall is there ever really a discussion of diet, of medication use, of other factors that could influence biomarker performance that's really unrecognized. And we have several other examples of how medication use, et cetera, can change these profiles. Nonetheless, there are a number of expression profiling studies in prostate cancer that clearly look like they could be attractive in terms of predicting outcomes. Here's one published by a group at Stanford. Here's a very recent one published by a group at Michigan that's looking at kind of somewhat stem cell markers, and this particular signature actually was capable of predicting outcomes in not only prostate cancer, but breast cancers and actually lung cancer. So you may see these signatures emerging into clinical use. However, very few of these actually have been validated in any sort of prospective manner. Now, there are some very exciting changes in the field that are starting to take advantage of DNA-based changes, which are very amenable to high sensitivity and high specificity type of tests. So this is a study by Rul Chinayan's group at Michigan that in many ways changed the paradigm of how we think about solid tumors and opened up an approach for diagnostic tests. So what this group actually showed is that, well, let me back up. It's been thought for many years that hematologic malignancies are quite different than tumors arising in solid organs, in one sense due to recurrent chromosomal rearrangement. So most of the hematologic malignancies, CML is a classic example, you have a recurrent consistent chromosomal rearrangement that's really the hallmark of the disease, whereas solid tumors were really thought to be characterized by much more random chromosomal events, rearrangements, breaks, amplifications, deletions, et cetera. So this paper was the first example in a solid tumor, in a carcinoma, where you have a recurrent chromosomal rearrangement. And what occurs here is a juxtaposition of a androgen-regulated gene, TMPRSS2, with a member of the ETS oncogene family, in this case ERG. So you now have an oncogene which was previously not regulated by androgen that now is regulated by androgen, which is an important hormone in the context of prostate carcinogenesis. It turns out this rearrangement occurs in about 50% of all human prostate cancers. To date, there's been some suggestion it may have prognostic value, but that's still not conclusive at this point. The attractive feature about this type of an event is it can be detected very sensitively by PCR. So that needle in the haystack issue that we were talking about earlier in the context of a protein-based biomarker can be circumvented by using PCR-based diagnostic methods. So if this fusion event, rearrangement event, actually turns out to have prognostic relevance, it'll be very, very useful. This group was also able to show that you could detect this fusion event, this genomic rearrangement in urine from men with prostate cancer. So there were enough prostate cancer cells sloughed in a urine specimen to be able to diagnose the presence of prostate cancer, at least the 50% of individuals that have this particular rearrangement. So what are the new approaches for discovery? How can we find additional markers that will be useful in this context? So I gave you one example of this DNA-based or gene-based method, biomarker that could be quite useful in prostate cancer. But I think where people have been trying to go is to go directly into blood proteins. The complexity, however, is extraordinary. I already talked about the dynamic range problem, but the complexity is another issue with modifications of these various protein forms. So this slide is simply meant to show that both the complexity and the abundance ranges are quite different when you're dealing with a protein-based marker versus a transcript or a gene-based marker. So the utilization of proteomics is now being widely studied by a number of groups, including our own, to try to identify protein-based changes in the blood that associate with the presence of malignancy or more aggressive cancers. So I'm not going to talk a lot about the details or the methodology of mass spectrometry, only to walk you through this slide without getting into a lot of the nitty-gritty details, which could take several hours in and of itself. So the simple concept here is to take a blood sample, extract the proteins, fragment the proteins, or digest the proteins into peptides. That can be then fractionated on high-performance liquid chromatography, then run through a mass spectrometer where individual peaks or representing not single proteins but groups of proteins or peptides can then be put through a second stage of the mass spectrometer where they're fragmented to generate what's called a fingerprint or an ion fingerprint. This fingerprint can then be searched against databases of known proteins or known peptides and then matched. So it gives you now a high throughput way of identifying proteins and their abundance levels in complex body fluids such as blood. Now you may be aware that several years ago there was a tremendous amount of hype of using proteomic-based methods for cancer diagnosis. So here's two examples, two papers, one looking at prostate cancers, another looking at ovarian cancers by using profiles of peptides or proteins in the blood. So you can see here's two unaffected individuals, and then the presence of these particular protein forms in the two individuals with cancer were thought to be pathognomonic of that disease. And the sensitivity and specificities reported in these studies were actually quite extraordinary. And it caused the field to question some of these reports. And this is a beautiful example of a well-written kind of rebuttal, if you will, to one of these papers in considering the realities of biomarkers. So what Dr. Diamandis actually showed here is to say, okay, here's your abundance levels of the abundant blood proteins that we've talked about before, albumin, immunoglobulins, et cetera. Here's the classic tumor markers and their abundance levels. So we're talking about things like PSA and CEA and that sort of thing. And yet in these papers, what was being reported were fragments of proteins, very common proteins, for example, haptoglobin alpha subunit, that were present in log orders of magnitude greater levels than these classic tumor markers. So it simply questioned what these proteins or pieces of proteins actually were, and how they would be produced by these tumor cells at these types of levels. Now, so what's happened is that reanalyses of many of those studies have actually demonstrated that they were, the vast majority, I won't say all, but the vast majority were due to artifacts and how the samples were actually handled. So I'm not aware of a single mass spec protein profiling study that was actually able to be validated and confirmed of having those types of sensitivity level and specificity level. So it's simply a word of caution that how these, what goes on in the front end with sample handling and sample collection is a major issue. Nonetheless, there is still a lot of investigation and work using top-down based proteomic strategies to try to define and identify biomarkers. And the basic concept here is you have some paired clinical sample and you evaluate those to determine differences. So you would compare serum or urine or CSF from individuals with and without disease. Now the major issues here, as I alluded to earlier, are the dynamic range and the relevance of those. And there are a number of possible solutions, and I'll touch on a couple of these. The first, there have been dramatic improvements in separating proteins into different fractions, and this is multidimensional protein separation using a variety of anion, cation exchanges, et cetera. So Sam Hanash's group at the Fred Hutchinson Center actually now does I think about 60 or 70 different fractionations before a given sample is actually even put on a mass spectrometer. Another is depleting the abundant proteins. So there are now very useful reagents produced by a number of companies that can deplete all of these very high abundant serum proteins. So you're left with basically that 1% of proteins that you want to evaluate for the candidate biomarker. You can also use methods to select proteins of a specific class, and what I'm showing you here is simply a glycoprotein. So Rudy Abersold several years ago at the Institute for Systems Biology developed a method to capture proteins that were glycosylated. These are proteins that usually end up on the cell surface or secreted, so you have an enrichment then for proteins that would be thought to be useful biomarkers. Now, we have a project at the Hutchinson Center at this point that's funded by the Allen Family Foundation to use mouse models as a kind of workhorse to try to identify blood-based biomarkers. I'm not going to go through all of this, but simply there are many advantages to using mice as a model system or a resource, and I'll give you one example here. So. This would, for example, be the tumor growth kinetics of a malignancy either in a transgenic mouse or a xenograft. So you're probably aware there are many transgenic mouse models now for just about all the major cancers affecting a man. This is a model of breast cancer where the HER2/neu oncogene is overexpressed specifically in the breast and is regulated by the addition of tetracycline. So you can either turn off or turn on the HER2 oncogene by the presence or absence of giving the mouse tetracycline in the drinking water. So you can see here on the far left, it's a fairly large tumor on the mouse, in the mouse breast. And by altering the tetracycline level over time, so here's just 21 days or 3 weeks later, that tumor is no longer visible. So you can regress it. By then manipulating the tetracycline again, you can cause that tumor to regrow. So you can imagine this is a great model system to be able to obtain blood samples over time as that tumor grows and then actually regresses. So you can have multiple time points from the same animal with high tumor burden and low tumor burden. It's a controlled environment, so you don't have to worry about diet, you don't have to worry about medications and that type of thing, and you can perform biological replicates. So this Allen Foundation project is incorporating several different mouse models of both breast and prostate cancer in this scenario, where we then evaluate both the tumor and the blood samples from these animals looking for biomarkers that would be present ultimately in the blood. And there is success here, at least in the mouse, and the future is to move this into humans then. So Mandy Paulovich, who's part of this Allen Foundation project, recently published a study of using this HER2 model to identify first protein changes in the breast tumor itself, and then to show that those changes also were found in the plasma. So there are several biomarkers here. You can see in the normal in the normal breast versus the cancerous breast. You can see from the back of the room the Western blots showing dramatic upregulation of these proteins that are present there, and then also in the serum or the plasma. And here's another marker, fibrillin-2, that here's a standard curve, and all of the cancer cases were found high up on this protein concentration in the blood, whereas the normal samples are down here. So there were several markers that emerged out of this study, initially from tumor profiling and then verifying that they were present in the blood. So to summarize and conclude, the validation of these markers is really where the rubber hits the road. There are a plethora of markers that have been identified from both tumor sampling and blood sampling studies that look promising, but validating them is really where the difficulty is. And that is determining, is this biomarker really associated with the target disease that you're interested in real world samples? And is that signal, is the difference in the benign or in the non-diseased individual strong enough relative to that underlying biological variation? And an important point to consider, so this is some more of Lee Anderson's work, is another factor that can clearly influence the utility of a marker. And what he did here is he took the most abundant plasma proteins from monozygotic twins, and simply their abundance levels could pair up 90% of these twins. So the abundance levels of these abundant proteins could pair up those twins, indicating that the genotype or the underlying genetics clearly matters simply in dictating abundance levels of common proteins. So I hope I've made the argument that early cancer detection offers the best hope at this point for reducing the morbidity and mortality of cancer in the population. Optimal screening probably will involve some type of a blood-based marker, although urine-based or other type of biospecimen analytes may also be useful. The current blood-based markers that we have for prostate cancer are really very poor. PSA has been out there for quite a while, but the operating characteristics of PSA still lack a lot in terms of sensitivity and specificity. We really need biomarkers in the prostate cancer area that can confer some idea of outcome or propensity for that tumor to grow and metastasize. And I would argue that may also be true for other malignancies. We just may not know it yet. If we start detecting a number of very small breast cancers, there may be a lot that actually never need to be treated. We just don't know it at this point. For prostate cancer, we've had the luxury, if you will, of having PSA to bring in men very, very early in the disease course, but a lot of those men actually don't need therapy. So we don't want to find these indolent cancers. We think that protein-based methods will offer the most direct route, but I think we're certainly not there yet. I showed you a few examples of plasma and tumor-based profiling. We hope that the mouse models, by having a high tumor burden, at least initially, compared to the organism weight or organism mass, will enrich for these, but that's still an open question. But the hope is that advances in these genomics and proteomics tools and the model systems and annotated biospecimen collections, which I think really has been underestimated and underinvested in terms of ultimately validating the markers. But I think these have some opportunities and optimism. And I'll end with this slide. So I mentioned that one of the active areas of interest for prostate cancer is a term called active surveillance. And that is that when men are diagnosed, they're not immediately treated, but actually followed closely with intent to treat if the tumor changes or tumor characteristics warrant it. So this particular project is one that's funded by the Canary Early Detection Foundation in partnership with EDRN. Right now it's a fairly small study of 5 institutions, and the plan here is that men are diagnosed at this point with a cancer that we feel comfortable watching, and they will have regular assessments over time with blood collections, urine collections, and serial prostate biopsies. If they reach certain trigger points, a more aggressive Gleason grade, for example, or the PSA, for lack of a better marker, going up very, very rapidly, then they'll go ahead and have primary therapy. So they'll have surgery, for example. So they'll continually be risk assessed. Otherwise, if they're not deemed to progress, then they'll continue on having these samples collected. So at the end of 3 to 5 years, we'll have a bank of well-annotated, collected materials in a consistent fashion that we can go back and evaluate any given biomarker, be it tissue-based, blood-based, urine-based, for its predictive power in determining cancers that do or are destined to progress. So I'll end here. There are a lot of people that have contributed to a variety of these studies. I'm not going to go through each of their names here, but I'd be happy to entertain any questions. Thanks.
Questions for Dr. Nelson? Go ahead,
Sure. Yeah.
So what evidence is there that early stage tumors, so the goal is to diagnose these tumors, solid tumors at an early stage. What evidence is there that early stage tumors actually release proteins and DNA into the serum? I mean, a pessimist might argue that if you're looking in the serum, you find anything, the cat's already out of the bag. So.
No, that's a great point. So in part it may depend on where that so it may not be true for every tumor type. One might imagine that the prostate or the breast or the lung may be more or less accessible in terms of the types of things that it may release. So for example, the prostate, you may be able to use the urine as an example. Lung, you may be able to use sputum or some issue in the blood. I think it's still a very open question of how early something might be shed. And there aren't great examples of being able to find subtle or protein alterations that would correlate with a very early presence of disease. I mean, PSA, as an example, one of the ideas is that you're actually changing the architecture of the gland. You're accelerating the release of that PSA into the bloodstream by breaking down the tissue-blood barrier. But it's hard really to know because PSA is a non-specific marker. So I absolutely agree. I think it's a very open question, and there aren't good examples that would prove the point at an early stage. I mean, some testes tumors, for example, even though they're localized, produce a large amount of HCG or alpha-fetoprotein without any evidence of metastases. So that might be maybe one of the best examples. I think something that is secreted, is actually produced in high amounts by the particular cell, intuitively might be the best type of candidate, and maybe that's why PSA is, it is present in reasonable concentrations in the blood. But I think the testes markers might be the best example of a small tumor still being able to put out a fair amount of material.
Other questions? Yeah. I thought that the study showing the dietary effect on transcriptomics was really quite interesting. And how were those specific genes chosen? Were they chosen in advance of doing the study, or was it sort of an after-the-fact analysis that showed that some genes had their transcript levels changed by diet?
Great. So the question was for the dietary study, were the genes that we saw altered, were those chosen a priori that we would investigate, hypothesizing that they might be changed to diet, or was it more of an ad hoc or after-the-fact analysis? And basically, what we had no a priori consideration of any particular gene that we thought might be altered by diet. This was a microarray-based study, and on the microarray there were about 10,000 or so genes. We did correct for multiple comparisons on that analysis of the pre and the post, and each patient was compared as his own control. And so those were the genes that fell out of that analysis. So I think the false discovery rate on that was about 10%. So 1 out of 10 of those genes is probably in error, but we have confirmed several of them by other methods like real-time PCR, so I'm pretty confident that there are significant changes that occur in that. Yeah.
Mark,
Yep.
another area of interest has been looking for circulating tissue cells. Where is that with prostate cancer?
Yeah, it's a great question. So there's a tremendous amount of work here looking at that with Bob Vasella and Paul Lang. So the studies are quite fascinating and very confusing. So it turns out most of their studies are looking in the bone marrow, since most men with prostate cancer, when they metastasize, it metastasizes to the bone. And so at the time of surgery, for presumed localized disease, so these are men coming in for surgical cure, they go ahead and do a bone marrow aspirate while they're under anesthesia, and then purify out epithelial cells using a couple antibody-based capture techniques. And what they can find is between 50 and 60% of all of these men with localized prostate cancer have detectable epithelial cells. I hesitate to call them prostate cancer cells or disseminated tumor cells. I can definitively say they're epithelial cells. Whereas controls, they have quite a number of controls, age-matched, et cetera. The numbers are about 5% or so. Barb Trask and Ilona Holcomb at the Fred Hutchinson, and we've done some work trying to characterize these cells, and a large number actually have chromosomal abnormalities. So very few of them express PSA. A small number do, but most don't express your classic markers of differentiated prostate cell, but the vast majority have chromosomal abnormalities. Almost 100% of the tumor cells captured from men, the epithelial cells captured from men with advanced disease, so men with known metastases, almost 100% of them have chromosomal abnormalities, and a large number of the men with only localized disease do. So it's an open question exactly. So there doesn't seem to be a prognostic, though simply having the presence or absence of cells in the bone marrow is not prognostic for who relapses. So we think that there's a subset of these cells that may have the signature or the phenotype to then establish a metastasis at some point in time, and that's an active area of research. But they can clearly be detected. I think a subset truly are neoplastic cells. It's hard to know at this point what that breakdown is.